Welcome to G Fun Facts Online. We tend to think of evolution as an incredibly slow, ponderous process, building new creatures over millions of years through thousands of tiny, almost imperceptible changes. And for the most part, that's true. But what if it's not the whole story? What if, sometimes, evolution takes a giant leap, all thanks to a single change in a single gene that fundamentally rewires the very architecture of the brain? Today, we're exploring the architects in the machine, the powerful master genes that have driven some of the most dramatic changes in the history of life. Let's start not with humans, but in the miniature world of the fruit fly, Drosophila melanogaster. The act of courtship for a fruit fly is not a simple affair. It's an elaborate, multi-part performance. A male fly must approach a female, tap her, extend one wing, and vibrate it to produce a species-specific love song. If his song is good enough, he proceeds to the next steps before mating. This entire intricate behavioral sequence is not learned. It is a hardwired instinct. And at the heart of this entire romantic drama lies a single gene, fruitless, or Fru for short. The Fru gene is a master regulator. Through a process called alternative splicing, the gene is processed differently in males and females. In males, it produces a unique set of proteins known as Fru-M. These proteins are transcription factors, which means they act as master switches, controlling a whole suite of other genes. Here's the incredible part. These male-specific Fru-M proteins are only active in about 2,000 of the fly's 100,000 neurons. These few scattered neurons form a dedicated circuit that governs every single step of the courtship ritual. During development, these proteins act as architects, sculpting the male brain. They guide neurons to connect in male-specific ways, essentially building the stage and writing the script for the fly's love life. If a male fly has a mutated Fru gene, he is anatomically normal but behaviorally lost. He fails to court females and will often indiscriminately court other males. But the story gets even more dramatic. Scientists looked at a different species of fruit fly, separated by 30 million years of evolution. This species doesn't sing. Instead, the male regurgitates a food droplet and offers it as a nuptial gift. They found that this species also has a Fru gene, but it was active in a small cluster of 16 insulin-producing neurons, where it was silent in the singing fly. This led to a stunning experiment. Using genetic tools, the scientists artificially activated the Fru gene in just those 16 cells of the singing fly. The result was astonishing. This simple rewiring was enough to create a new hybrid behavior. For the first time, the singing fly males began to perform the gift-giving ritual of their distant cousins. It was the first time a complex, species-specific behavior had been transferred from one species to another by manipulating a single gene. This shows that evolution doesn't always need to build a new brain from scratch. Sometimes, just changing the expression of one master gene in a handful of cells is enough to create a dramatic new behavior. Now, let's scale up from fruit flies to the 3-pound universe inside our own skulls. The evolution of the human brain, with its capacity for language and abstract thought, presents a challenge of a different magnitude. What genetic changes could account for the vast cognitive gulf between us and our closest relatives? While the full answer involves many genes, researchers have identified a dramatic, single-gene event that appears to have played a pivotal role: the duplication of a gene called SRGAP2. About 3.4 million years ago, in the genome of one of our hominin ancestors, a segment of a chromosome was mistakenly copied, creating a partial duplicate of the SRGAP2 gene. Then, around 2.4 million years ago, another duplication happened, creating SRGAP2C. This timeline is tantalizing, as it corresponds roughly with the transition from the smaller-brained Australopithecus to the genus Homo. a period of rapid brain expansion, and the first appearance of sophisticated stone tools. What's special about this duplicated gene is what it does. The original gene, SRGAP2A, acts as a brake on brain development. It tells developing neurons to mature quickly, limiting the number of connections, or synapses, they can form. It says, okay, stop growing and start working. The duplicated copy, SRGAP2C, is an incomplete version. It can still bind to the original protein, but it can't do the job. So it gets in the way, effectively interfering with and weakening the stop signal. The result is a phenomenon known as neoteny, the retention of juvenile features into adulthood. With the brakes weakened, our developing brain cells stay in a more immature, plastic, and exploratory state for a much longer time. When scientists introduced the human-specific SRGAP2C gene into the brains of developing mice, the results were incredible. The mouse neurons took longer to mature, and because they had more time to grow and explore, they ultimately formed a much higher density of complex connections. They became more richly wired, mirroring a key feature of the human cortex. This single gene duplication, a simple copying error, seems to have lifted the brakes on our neuronal development, allowing our brains to become more densely interconnected and paving the way for our unique cognitive abilities. Of all our abilities, complex spoken language is perhaps the most uniquely human. For decades, the search for its genetic roots was a holy grail. The story famously broke open in 2001 with the study of a large family in London, known as the KE family. Half of its members suffered from a severe speech and language disorder. They struggled with grammar and the complex motor control of articulation. Researchers discovered the cause was a mutation in a single gene, FOXP2. FOXP2 was sensationally dubbed the language gene, but the reality is more nuanced. Like fruitless, FOXP2 is a master regulatory gene, a transcription factor that is critical for the normal development of the neural circuits that support speech and language. The human version of the FOXP2 protein differs from the chimpanzee version by just 2 amino acids. While the evolutionary story of when these changes became fixed is still debated, their functional importance is becoming clear. To figure out what these human-specific changes do, scientists engineered mice to carry the human version of FOXP2. Of course, the mice didn't start reciting Shakespeare, but they did show significant changes in their brains and behavior. Specifically, neurons in a part of the brain called the basal ganglia, which is crucial for learning motor skills, became more complex and showed changes in their ability to form and strengthen connections. Most importantly, these humanized mice became significantly better at procedural learning. This is the type of learning where you turn a conscious effort, like learning to ride a bike or type on a keyboard, into an automatic, unconscious routine. Fluent speech is a marvel of procedural learning. We don't think about the hundreds of precise muscle movements our tongue, lips, and larynx make. The human version of FOXP2 seems to have tweaked our brains to be better at automating these complex motor sequences, a critical skill for fluent speech. It didn't create language, but it appears to have laid some of the essential neural groundwork. These stories are just the highlights. The genome is filled with other examples. Genes like ASPM and microcephalin, when mutated, cause drastically smaller brains. Their evolutionary history in the primate lineage shows strong positive selection, suggesting changes in these genes helped drive the massive expansion of the human cortex. Even the domestication of animals might have a simple genetic basis. The domestication syndrome, floppy ears, patchy fur, and reduced fear, is now thought to be linked to genes that regulate a population of stem cells called neural crest cells. By selecting for just one trait, tameness, our ancestors may have inadvertently selected for a mild deficit in these cells, which had a cascade of effects on everything from coat color to the adrenal glands that control the stress response. Together, these discoveries are changing our view of evolution. It isn't just a slow, gradual march. It also works in leaps and bounds. Single genetic events, a duplication, a mutation, a change in expression, can act as powerful architects, creating major new blueprints for natural selection to work on. These single genes sit at the top of a pyramid, directing a whole network of other genes to rewire the brain. Deep within our DNA, within the code of these single powerful genes, lie the stories of the great leaps that rewired our brains and, in doing so, made us who we are.